Hello, everyone. Welcome to today's podcast. This is for day 17, and this covers bits and pieces from chapters 12, 17, and 18. I'm going to go a little bit out of order, and I think watching the podcast for Objective 2 is the best approach, and then we'll come back to Objective 1 and 3 for the second set of podcasts. This Objective 2 focuses, as we can see here, on mutations and how they occur and what's the consequences of those mutations. When we get done with that, we'll move into back to Objective 1, where we talk about the cell cycle and how the cell cycle is set up in such a way to prevent mutations. And then we'll close with a discussion on cancer, because that then leads us into what happens when the cell cycle doesn't do its job and allows mutations to occur, and then of course that can lead to cancer. But we're going to start with Objective 2 here, mutations, and we're going to talk about defining the mutations, the types of mutations there are, and how it affects the protein, and then talk about different ways that these mutations are actually caused. All right, so let's go ahead and start with the term mutation, and let's define it. A mutation is nothing more than a change in DNA sequence. So let's go ahead and write that down here too. Change in DNA sequence. Now, to illustrate what a mutation is, let's go ahead and just write a generic DNA sequence here. And I'm going to put non-mutated sequence here, so non-mutated. I try to avoid using the words normal and abnormal, because sometimes those terms aren't appropriate to use. In fact, a lot of times they're not. So we're just going to use non-mutated. And let me go ahead, and I'm going to write the non-template strand here. And let's begin with ATG, GTC, TAC, GAT, AGA, and let's say that this is a very long gene, and so let's say there are 500 more codons here, and I'm just not going to draw them off because we'll be here all day if I do that. So there's a lot more. This is a huge sequence, and eventually it's going to end with a stop codon. And so let's pick UAA. Now, we can mutate this DNA sequence in several different ways, but let's first start with with a substitution. So let's write substitution here in blue. And I'm going to write the rest of the sequence all in black except for the one substituted a base, and I'll explain what that does in just a moment. So let's go ahead and I'm going to back up real quick. The reason I chose to write the non-template strand is because we can look at this, and just by substituting the T's for the U's, we can use this directly and look at the genetic code, and in a moment we'll do that and be able to tell what proteins are being made, or what amino acids are being coded for by this. All right, so ATG is going to be unaffected. Same thing as GTC, same as TAC, and we're going to get a change here in GAT. Instead of GAT, we're going to substitute that A for a T. So let's put a blue T here. All right, and then the rest of the protein is not affected. We still have our 500 codons here, followed by the stop codon of UAA. All right, so a substitution typically is not as severe as the next two we're going to talk about, an insertion or a deletion. And that's because all we're doing is changing this one codon. And so when we start translating these into proteins, what we're going to discover is that what we've changed here is just one amino acid. And we'll come back to that in a moment. I don't want to talk about that quite yet. All we change is one amino acid, or another way to think about it is that we've changed one word in this sentence that we call the protein sequence. So just one amino acid has been changed. So often that doesn't have an effect. Now, sometimes it can have a drastic effect, but usually they're not near as severe as an insertion or a deletion. So let's talk about a deletion first. What do deletions do? And when we talk about a deletion in this context, we're only going to talk about removing one nucleotide. And so let's say ATG is fine. But let's say C has been mutated. And so now we don't have any base here. We've crossed off C. It's gone. The rest of the nucleotides remain unchanged. So what effect is this going to have? So remember, our genes, our DNA sequences, don't know that these three are supposed to be together. It just reads three at a time. So it's going to interpret this one as our ATG. Then it looks for the next three. So it's going to take this GT. There's nothing there, so it's just going to look for the next one. So the next codon now will be a GTT. And the next one will be ACG. The next one will be ATA, GA something here. And then all the way down. And so what you see, what happens here in this deletion is we still make our first one, and we'll see in a moment that that's methionine. But now we no longer make this one, GTC. We now make GTT. And that changes it from a valine, GTC, to a GTT, which is still a valine. So we're actually in good shape there. Now let's go to the next one, ACG, which is now threonine. So that's changed now. And what you'll see is we got lucky on the first one, went from a valine to a valine, but every other amino acid now changes all the way down. And eventually what you'll probably hit is a premature stop codon, or it won't read this stop codon anymore and it'll make a much larger protein. Either could occur. We call this a frame shift. And so I'm going to write the word frame shift here. The reason we call this a frame shift is that what we have here, like in our normal sequence, this is our reading frame. And by removing one of the nucleotides, we've changed the reading frame. We've shifted it down one nucleotide, so we call it a frame shift. And as you can see, it changes every amino acid after that. Every codon after that changes, and then every amino acid changes. Let's back up a little bit. It doesn't necessarily change every amino acid because we coincidentally just got lucky on this first one, but it almost always changes every single amino acid that follows. These, because of that, tend to be very dangerous because now you're making a protein that is completely different than what was intended here. Whereas the substitution, you just changed one amino acid. Usually not as severe. Now, while we're going to talk about insertions in a moment, but while we're on this train of thought, sometimes it depends on where that frame shift occurs. If it occurs at the very beginning of the protein, of the gene, then that means every nucleotide after it is affected. Every codon afterwards is affected. So usually a deletion in the beginning of the gene is more detrimental to the cell than one that occurs near the stop codon, because you've still made almost that whole protein, and it's that whole gene has been unaffected. Okay, so the placement of the deletion early in the gene or late in the gene can also have an effect. So let's talk about an insertion, and we'll make it in green. Okay, the insertion is when we take a nucleotide and insert it into the reading frame. And what we're going to do is we're going to insert a nucleotide. If we look up here, the normal one, we're going to insert one right here when we get to it. So let's go ahead and write the sequence. And let's insert a G right here. And the rest of the sequence is going to remain the same. Remember, we didn't lose the C, so we have to keep the C. C. GA, TAG, A, and then several, and then a UAA. What you might have noticed is insertions also create a frame shift. We have these three aren't affected, these three aren't affected, and now instead of a TAC, we have a TAG. We inserted a base here. Now the next one is going to be CGA. Before it would be a GAT, so we've changed that codon. Then the next one is TAG instead of AGA. Again, we've changed that one, and we're going to change every single codon afterwards, all 500 of them here, until we get to this stop. And again, we may blow right through this stop here because of the frame shift, or we may encounter an earlier one. Now I specifically chose this one here, this TAG, because TAC, our original one here, if you look at your genetic code, and in fact, I would recommend go ahead and opening up your textbook so you can look at the genetic code while you do this, because that's going to come in handy now and also in a little bit. So go ahead and hit pause and pull out a genetic code. As you did that, TAC, let's see what that makes with the TAC. You're going to want to look at, remember, replacing all the T's with U's when we're making this translation. So we're looking at a UAC. A UAC, if you look at your genetic code, is a tyrosine. And we just made it into a UAG, and a UAG is a stop. So sometimes you can have these drastic changes, as we saw with the deletion in the frame shift. In this case, this frame shift caused a premature stop codon. So this, of course, is also going to be rather deleterious to the cell, because it's going to lose this entire protein. Okay, before we move on to the next slide, go ahead and hit pause here in a few seconds and go ahead and translate each of these pieces of DNA, the unmutated one and the various mutated ones, and see which amino acids are being produced. Remember, as you do this, you can just look at this non-template strain. You don't need to make the messenger RNA from it. Just as you look at the genetic code, replace the T's for the U's, and just look at those three bases. Fill that out, and then you'll be able to see how this changes the protein that is actually being produced. All right, now I want to spend a little bit more time talking about these substitutions. Remember, on the last slide we said the substitutions only change one amino acid. And sometimes that can be a problem, sometimes it's not a problem. It's typically less of a problem compared to the insertion or deletions. So let's introduce some terms for you here that help us understand how substitutions can have different effects. And the first one is nonsense. A substitution can be called a nonsense mutation. And a nonsense mutation changes a codon that codes for an amino acid to a codon that codes for a stop codon. So it's usually a more severe substitution here. So for instance, if you had a UAC and you change the C to an A, you would now have a stop codon. So creates stop codon. Let's write that here. Creates stop. And an example is UAC. Well, and then remember the DNA, this is going to be a TAC. A TAC in the DNA gets changed to a UAA or TAA in the DNA. Remember, it's the DNA that gets mutated. RNA doesn't get mutated. It's just the DNA. So this is a nonsense mutation. Now, depending upon where this nonsense mutation occurs, will decide whether or not it has a big effect on the protein that's ultimately made. If this occurs, if you have a 100 amino acid protein and this occurs at the second amino acid, usually this is bad news, right? This truncates most of the protein except for the first one or two amino acids. Now, if the nonsense mutation occurs at the second to the last amino acid of a 500 amino acid protein, then it's not going to have as big of an effect. So location is really important in deciding whether a nonsense mutation is going to create a problem. Okay, now let's think about a missense mutation. A missense changes the amino acid. And for amino acid, I'm just going to write A period A. This would be like, let's look for a good example down here. And let's look for an example that's actually going to have a larger effect. So for instance, aspartic acid here. This is a GAC, so let's write GAC here. So this is what's in the DNA, and when it's transcribed, it will make, and then translated, it will make aspartic acid. Now you don't need to know this for the exam, but to illustrate the example here, I'm going to tell you that aspartic acid has a negative charge to it. Now let's say we change it to a GCC. So we replace that A, substitute that A for a C. So now we go from a GAC to a GCC. And I can tell you, and again, you don't need to know this part for the exam, but alanine is non-charged. It's a non-polar amino acid. So if you think about this, we've made this amino acid less charged. We know from other examples that a negative amino acid is often found in the inside of the protein, where an uncharged one is on the outside. I'm sorry, I said that backwards. A negatively charged amino acid is on the outside of the protein, whereas the nonpolar one's on the inside of the protein. So now we've set up a situation where we may now have a protein that's going to fold differently. Now the next thing I want to talk about is missense mutations that aren't quite as bad as this. Okay, so let's talk about other kinds of missense mutations that change the codon to an amino acid that is less severe. And the first one here that we can talk about is a silent mutation. So let's write silent. A silent mutation is one where you change the codon, but it doesn't change the amino acid. So for instance, because of the redundancy of the genetic code, we have several codons that code for leucine, 6 here. We have 4 for valine, we have 4 for proline, 4 for serine. And typically what you'll notice is if you change the last nucleotide, you don't change the amino acid. So these are the third position. If you're going to get a mutation, usually that's the one you want to get because it's less likely to change the actual amino acid. But let's look at an example here. And let's just look at, arginine over here. Okay? So you could have let's say no change to amino acids. So let's put that here so we don't forget what it actually is. No change of amino acid. An example of this would be changing an AGA to a AGG. So this third position changed from an A to a G, and you still have an arginine. So here are 2 arginine codons. So we call that a silent mutation because the protein that's being made is going to be the exact same. Now the last one is called neutral. So a neutral mutation, let's define that here. Amino acid change, it has the same properties. Let's call it properties. Same properties. And an example of that would be something like leucine, CUG, changed to GUG. So leucine to valine. You don't necessarily need to know the categories of each of these amino acids. You don't need to know that leucines are hydrophobic and lysines are basic. You don't need to know that. But what you do need to know is that a neutral change is a change of amino acids in the same category of amino acids. So a positively charged amino acid to a positively charged amino acid. Okay. Valine to leucine. This is a nonpolar to a nonpolar. If you look these up, you don't have to, but if you were to look these up, you would see that the side chains of a leucine and a valine are very, very similar. It would be really unlikely that having just this one change, one leucine to a valine, is going to have a very minimal, if any, impact on that protein. So it should not have any change on the protein. So we call it a neutral change. Okay, let's go to the next slide real quick. And let's talk about how mutations occur. And we don't want to go into great detail here, but we should introduce this topic. Mutations can occur in one of two ways. The first way I'm going to indicate here are by mutagens. Mutagens are chemicals or radiation that cause DNA mutations or causes mutations. These are things that we're exposed to in our environment. Some of these are byproducts of our own metabolism, but let's go ahead and put, think a couple examples here. X-rays would be a good example. UV light is another really good example. Various chemicals. These could be chemicals found like in the workplace. They could be chemicals found at home, but they are all mutagens. And the last one, which is a really important one, is sometimes viruses, actually a lot of times viruses, can cause mutations. Another important way that mutations occur, and we're going to call these spontaneous. Spontaneous mutations. These occur during the cell cycle, and we're going to talk about this on the next podcast quite a bit, so I'm not going to spend too much time with it. Just recognize that as DNA is being replicated, the enzymes that are involved in that, they're really good enzymes. They very, very rarely make a mistake, but they're not perfect. Every now and then, when they're replicating the DNA, a mistake will occur. So we call these spontaneous, and they're the result of the cell cycle. And we'll talk a lot more about that in just a moment. Now, one last thought here. I want to just make sure we're clear on something. Often, when we hear the word mutation, we think of a mutation as being harmful. Let me make a better arrow here. Harmful. Now, it is true that some mutations can be harmful. Some can be lethal. However, some mutations are actually beneficial. These beneficial mutations are the source of generating diversity within populations, within communities. So sometimes mutations are beneficial. These are the reasons that organisms evolve, because of these beneficial mutations. A mutation that gives that organism some kind of competitive advantage. So I want to just make sure when we hear the word mutation, we don't automatically assume it's bad. Okay. Now, some of them, as we saw earlier, are neither. Let's just put neither. They're neutral. That would have been a better word to write, but I'll stick with neither. These mutations are neutral. They don't have any kind of effect. Maybe it's because the changes from a valine to leucine, like we talked about on the last slide. Or, remember, sometimes mutations occur in regions of our genome that don't seem to have any impact on us. Less than, somewhere between like 1 and 5% of our genome codes for proteins. That part of our genome, if you get a mutation in it, it's more likely to be harmful or beneficial. But well over 95% of our genome doesn't code for a protein. Doesn't mean it's not important. It just means it doesn't code for a protein. A lot of those mutations don't have a harmful or a beneficial impact. So a mutation could be one of these three things. That's just something good for us to keep in mind. All right, well, that ends this objective. If you have any questions, please make sure you come see one of us. Tune back in for the next objective, and we're going to go back and cover objective 1 now. Okay, bye.